Good morning. Why don't we go ahead and get started? Can you hear me okay, Gabe, on that end?
Yes.
Okay, great. Well, I hope everybody is safe and healthy. Thank you so much for coming this morning. Of course, it's Gabe Velez's Defense Day. We'll have about an hour Zoom here where Gabe will give his presentation. And then afterwards, we have a separate Zoom invite for his thesis committee that will go to. I've known Gabe, I think it's a little over 5 years now. It's been an incredibly exciting time. I think I feel a lot older, but Gabe mostly looks the same to me. He's certainly grown and matured as a scientist. He's had some really exciting work accomplished in the laboratory that he'll be focusing on today. When he started, he was very computational to begin with, and he's as good as his computational skills are. He's now extraordinary at the laboratory bench. Gabe is supported by an NIH F series grant. He's also contributed to a number of other grants in the lab that have gone on to be funded. Certainly, what distinguishes Gabe from uh, everybody else, quite simply, is that he's had over 35 peer-reviewed publications, many
36.
of them 36 now. So, and many of those first author, but I think equally important are the ones where Gabe is a second author or middle author, where he's really demonstrated his ability to jump onto a variety of projects, generate important experimental data, and convert that into meaningful publications. As part of the MD PhD program, it's our hope that we train somebody who's eventually going to have an impact on human health and to bring the skills of a scientist to that. And I think there's not much more we could ask for somebody like Gabe coming to that point. Of course, he's not going to get a chance to talk about all 36 of those papers, but he'll be sharing some of the really exciting structural biology and proteomic type of data that's given us insight into a rare eye disease. And I think Gabe's work and reaching this point and all his friends and colleagues coming together this morning here on Zoom should be a matter of pride for everybody. It certainly is for me. I think he's represented the program so well during his time out here at Stanford and integrated so nicely on the campus here. And I think his family, friends, and the University of Iowa should be incredibly proud of him. And for a son of Puerto Rico to come this far, it's just been an extraordinary privilege for me. So I'm going to let Gabe take over. I'm going to mute myself.
Okay, I'm gonna go ahead and share my screen, and just give me a thumbs up if you can see my screen here. Okay. All right, good. All right. So thank you, Vinu, for the introduction and those kind words. And I also want to thank the Graduate College and my thesis committee for being so flexible and allowing me to present my thesis seminar over Zoom live from my bedroom. I'll be going over some of the work that I've been doing during my PhD, and I've titled the talk Structural and Proteomic Insights into the Pathogenesis of Intraocular Inflammation. So I guess Vinu doesn't really need much of an introduction, but it goes without saying that it's been a pleasure working at the Byers Eye Institute in the Mahajan Lab at Stanford. As many of you are aware, I followed Dr. Mahajan in his laboratory 3 years ago when he took a position at Stanford, and I've been spending those last 3 years completing my PhD thesis work in the Bay Area. And my PhD is in molecular medicine. And with that, we'll get started. So when I first started my PhD, I became interested in the study of proteins. And I became interested in the study of proteins on two different levels. One on the order of a single protein, where we study its structure and how that unique structure guides its molecular function. And on the order of the entire proteome, which encompasses the large-scale study of protein expression and its modifications in the context of a biological sample. And this interest prompted me to join the Mahajan Lab, where we study protein structure, function, and expression in the context of vitreoretinal diseases. And while I don't have time to go over all of the projects that I worked on during my PhD, I will be going over a few projects that I feel encompass both of those areas of research quite well. I'll be discussing a rare inflammatory eye disease, and then we discover the protein that was responsible for the disease, and I'll be discussing its structure and its function. And then I'll be discussing its downstream signaling pathways that we discovered using proteomics, as well as therapeutic drug repositioning as a precision medicine strategy to treat these patients. And with that, I want to start off just by presenting a case. So a few years ago, my PI saw a patient at the University of Iowa. This was a 23-year-old male with slow vision loss in both eyes. And the eye is like a petri dish. And if we shine a light into the back of the eye using a slit lamp, we can start to count some of these clumps of white dots. These are clusters of leukocytes, and they're a sign of inflammation inside the eye. And this inflammation is referred to as uveitis. And most of the time, we don't know why it's happening or if there's a systemic underlying condition that is contributing to this inflammation. But other than this patient's eyes, he was totally healthy. And if we dilate the pupil and look into the back of the eye, we can take a look at the retina and examine it. The retina is like film on a camera. And this patient had scar tissue forming over the retina, and it was causing wrinkling of the retina, and that was distorting his vision. And that's another sign of inflammation. And amazingly, his mother was sitting next to him, and her eyes looked like this. One eye was filled with blood due to a vitreous hemorrhage that was blocking her vision, and the other eye was dead due to years of chronic inflammation. This is an eye that has been on fire for about 50 years, and doctors and healthcare teams haven't been able to put out that fire. And to see intraocular inflammation in related family members is quite unusual because uveitis tends not to run in families. And this mother and son were actually part of a large Iowa family that has been affected by this disease for over 7 generations. And most of the people that have been affected by this disease go blind in their 50s. And this is the Jackson family. They were featured in People magazine. And many of these family members are patients at the University of Iowa. The disease was described clinically and was called autosomal dominant neovascular inflammatory vitreoretinopathy. And that's a bit of a mouthful, so we'll call it ADNIV for short for the duration of the talk. But the name actually describes the clinical phenotype quite well. Autosomal dominant referring to the inheritance pattern, neovascular referring to the bleeding that occurs inside the eye, and vitreoretinopathy referring to the scar tissue that forms over the retina. The disease itself progresses in stages. The patients are usually asymptomatic for the first 20 years of life, but in the second decade, they get inflammation in T cells similar to uveitis. In stage 3, they get angiogenesis and vitreous hemorrhages, as well as neuronal cell death, and this looks a lot like diabetic eye disease. And in stage 4, they get intraocular fibrosis and retinal detachment, and this looks a lot like a surgical complication called PVR. And it's as if these patients are developing several distinct eye diseases, but in different stages. And we see these eye diseases all the time in the eye clinic, but it's rather unusual to see them occurring in the same patient sequentially. And for this reason, we refer to ADNIV as a Rosetta Stone disease. And we're interested in the lab as in uncovering the mechanisms underlying ADNIV so that we can get some insight and into treating ADNIV patients, but as well as insight into more common causes of blindness. So when our lab first performed whole exome sequencing on 3 different ADNIV families, they found that they all harbored point mutations in the CAPN5 gene. This encodes for a calcium-activated cysteine protease called calpain-5. I've shown a homology-based model of the protease core domain here. It contains two globular subdomains termed PC1 and PC2, separated by a flexible linker. And the active site groove is shown in the middle here. And I like to imagine calpain 5 as a bit of like a Pac-Man-like molecule. When it's activated by calcium, these two lobes come together to facilitate formation of the catalytic triad. And interestingly, our first identified patient mutations were located on this regulatory loop here, termed the G1 gating loop. And this loop has been shown in other calpains to block the formation of the catalytic triad and block access of substrate into the active site in the absence of calcium. So we believe that these patient mutations disrupt this regulatory feature and allow the protease to be active even in the absence of calcium. So they would result in hyperactivity. Calpain 5 is a member of a large family of cysteine proteases. There are 15 total in humans, and all of them contain this protease core domain, and they can be grouped into classical or non-classical calpains. Calpain 5 is a non-classical calpain and is quite divergent from its classical counterparts in terms of its domain architecture. What's most striking is that it lacks this penta-EF hand domain, which is a regulatory domain involved in calcium binding, as well as binding to the small subunit and the endogenous inhibitor calpistatin. And I'll get back to that in a couple minutes. But there is this precedent that calpain 5 hyperactivity would be associated with retinal disease because calpain hyperactivity in general is associated with multiple disease processes, such as stroke, heart attack, reperfusion injury, and retinal degeneration, to name a few. And the underlying idea here is that one of the consequences of tissue injury or ischemia is that you get elevated intracellular calcium, and this leads to aberrant overactivation of calpains in the cell, leading to proteolysis and cleavage of cytosolic, nuclear, and membrane-bound substrates, which then leads to altered cell signaling and cell death. And for this reason, there have been a number of efforts to inhibit calpains in disease. There is, in fact, an endogenous inhibitor of calpains in the cell called calpistatin. It's a 700 amino acid long polypeptide that sort of lassos around the active site, as shown here, and docks its N and C termini at the penta-EF hand domains, which are critical for maintaining this interaction. But since calpain 5 doesn't have these penta-EF hand domains, it's not believed to be regulated by calpistatin in vivo. There have been several non-specific active site inhibitors that have been developed, like lupeptin and several alpha-ketoamide-based molecules, but these are rather unspecific because they inhibit all calpain isoforms, as well as several families of cysteine proteases. So they would be too broad and non-specific for clinical use. So we're left with this problem that we don't have a calpain 5 specific inhibitor, nor do we have a high resolution structure of calpain 5 in order to guide rational drug design. And with that, I'll be going over a paper that we recently published in Cell Reports describing the structure and the function of the calpain 5 protease core. So for this study, we focus on the protease core domain because it's easier to purify recombinantly and work with in vitro than the full length, and it contains most of the known additive mutations. So I've shown here a sequence alignment of calpain orthologs, highlighting the positions of known disease mutations in relationship to functional residues like the catalytic residues shown here. And for the duration of the talk, I'll refer to this region of the protein as calpain 5PC. So for the non-structural biologists tuning in, there are many different ways to solve the structure of a protein. Our lab used X-ray crystallography in collaboration with the protein and crystallography facility at the University of Iowa. And briefly how this works is we express our protein of interest in bacteria, we use E. coli, and we grow up these cells in several liters. We then lyse them and purify them through a series of affinity and size exclusion chromatography steps until we get recombinant calpain 5 that's pure and sufficient for structural studies. And once we do, we can then screen growth conditions to see which conditions will give us crystals of our purified protein of interest. And once we do get crystals, we can take them to a synchrotron radiation light source where we perform X-ray diffraction experiments. We're fortunate enough to have two of these facilities in the Bay Area, the Stanford Synchrotron Radiation Light Source and the Advanced Light Source at the Lawrence Berkeley National Lab. And here we take our crystals, we'll mount them in front of an X-ray source and shoot them with high energy X-ray beams. And if the crystals are well ordered, and a little bit more importantly, if we're lucky, they will diffract. And we can measure this diffraction on a detector and collect this data. And with a few bits of other data that we measured indirectly, we can solve the structure of our protein of interest. Now, I want to stress that this is a rather labor intensive process. So, oftentimes we're screening thousands of crystal growth conditions to see which ones give us hit conditions. Um, Oftentimes, our hit conditions look a lot like this. We'll have these like thin, very fragile crystals that really are too small to diffract on a synchrotron light source. And we'll go through several iterations of optimization in order to get crystals that are much bigger and look something like this. Sometimes we'll get lucky and we'll get these beautiful looking crystals, and I'll take pictures of them and show them to my PI. And sometimes there's some premature celebration. We have crystals, which is very exciting, but then I'll take them to the synchrotron light source and I'll do X-ray diffraction experiments, and our diffraction data will look a lot like this most of the time. We'll see these a few reflections here that are located far away from the beam stop, and this just tells us that we've successfully crystallized salt, which is unfortunately pretty common. So this is an effort that has required a lot of effort from members of the Mahajan lab as well as the protein and crystallography facility. But after several months of attempts, we've been able to crystallize the Capn5 protease core structure to a resolution of 2.8 angstrom and solve the structure. You can see that it retains this globular PC1 and PC2 subdomain architecture, and I've highlighted the location of the active site as well as that G1 gating loop where the original additive mutations were originally identified. If we zoom into the active site, we can actually observe the catalytic triad. You can see here that this tryptophan 286 residue is forming pi-pi stacking interactions with the catalytic histidine, and this is blocking formation of that catalytic triad. This is another regulatory motif that prevents aberrant calpain activation in the cell. We can observe downstream at the hydrophobic core and see that calpain-5 has a stable alpha-7 helical region, and this provides some additional structural support to the catalytic triad near the hydrophobic core. And there have been several calpain structures that have been solved and deposited in the PDB, and some of them, like human calpain-1, lack this helical region, and they undergo hydrophobic core collapse in the absence of their non-catalytic domains, and are therefore weakly active or inactive in vitro. So we hypothesize that because of this increased structural stability near the hydrophobic core, that calpain-5 PC would be active in vitro in the absence of its non-catalytic domains. So we decided to test this hypothesis using a fluorescence-based enzymatic activity assay. At the time that we were doing these assays, the natural substrates of calpain-5 were not really fully characterized. So we used synthetic substrates known to be cleaved by other calpains or cysteine proteases in order to measure calpain-5 activity in vitro. This is a diagram or representation of one of these substrates. You can see that it's an N-protected tripeptide with a fluorophore conjugated to it and a quencher. And when this peptide is intact, this quencher will then quench the fluorescence. When calpain-5 comes in and cleaves it, this fluorophore is released, and we can measure activity as a continuous readout of fluorescence on a plate reader. So this is an example of one of those reactions where we see an increase in fluorescence in the presence of calcium. When we mutate that catalytic cysteine to a serine, we see a characteristic loss of activity, and this is just telling us that our calpain-5 protease core is behaving like we would expect in vitro. And we were able to use these assays to map the kinetic parameters as well as determine some of the calcium requirements for calpain-5 activity in vitro. But what was really important to us was understanding how calpain-5 PC was unique to other calpain cores. And the reason why we wanted to do this and understand this is because if we could identify unique features or sites on the calpain-5 PC molecule, then these could be served as sites for rational drug design. So when we're developing a small molecule inhibitor, we want it to be as specific as possible. There are many calpains in the cell, and they all perform unique physiological functions. So to inhibit all of them would lead to a wide side effect profile that's rather undesirable. So one of the ways that we can assess the functional diversity of calpains is through sequence-based phylogenetics. And here the idea is that we take known calpain sequences and they'll undergo multiple sequence alignment in order to reconstruct these large phylogenetic trees where we can infer the evolutionary relatedness of different calpanes and identify some unique sequential features of calpane 5 compared to its classical counterparts. But in this world of genetics that we live in, we sometimes lose some of this resolution that we're looking for in terms of structure-based drug design. There are some features that you can't necessarily assess in one dimensions, and you have to visualize them in 3D in order to get a better sense of how these molecules are differ and how they're similar. So I worked on developing an informatics pipeline to assess the functional diversity and differences between 3D shapes. I'll go over a little bit how this works. Basically, we will download all the calpane structures that have been deposited in the protein data bank. We will extract their protease core domains and filter them by several X-ray crystallographic quality control metrics. We then feed it into our analytical pipeline and align these PC structures by their alpha carbon atoms. And this allows us to calculate a root mean square deviation or a distance in angstrom between the two overlaid pairwise structures. And we'll do this for all the different combinations of structures in our pool and generate a structural dissimilarity matrix that we can then feed into basic phylogenetic algorithms to reconstruct a tree that is just based on the structural differences alone and not based on sequence. And from here, we can identify different clusters of related proteins and identify unique structural features on CAPN5. And with the help of Jing, a research assistant in our lab, we've been able to automate this process and turn it into a program that we currently call Philo3D. And hopefully this program will be available online for public use in the near future. So this is the result of our analysis. A sequence-based phylogenetic tree will infer the evolutionary distance of molecules based on their differences in the multiple sequence alignment, but a structure-based phylogenetics tree will infer those differences based on how similar the structures are in 3D space. You can see here already that our CAPN5 PC1 substructure is divergent from its classical counterparts, and we can already start to identify some of these unique features, such as this long flexible loop that was absent in classical calpane cores that we called the PC1 L1 loop. If we look at calpane PC2 substructures, we can identify even more unique features. This one is a little bit more divergent from its classical counterparts. We can see that it has this long alpha 8 helical region, as well as several elongated beta strands that we weren't able to see on sequence-based phylogenetics alone. And we also identified this long PC2L1 loop and this long PC2L2 loop that were absent on classical calpane structures. Next, we map the location of known adnith-causing mutations onto the 3D structure. And one of the interesting mutations that we identified was this G267S mutation. It was interesting because it was not found on any of the classical calpane structures, only on calpanes 5 and 6, which are in the same subfamily of non-classical calpanes. So it appeared to be unique to these non-classical calpane family members, and it was located far away from the active site. So you can see this loop here, and the active site is all the way down here. So we were uncertain how this mutation that was located far away from the active site could confer hyperactivity downstream. And what was more interesting was the patient phenotype. So this was a patient that was seen by one of our colleagues at Oxford University. All of the previously identified ADNIV patients that we had described were asymptomatic until they were in their 20s, but this patient actually had visual problems since infancy, and they had recurrent severe vitreoretinopathy and retinal detachments. You can see their fundus here. There's a lot of scar tissue and vasoproliferative pathology. So we had a very aggressive phenotype associated with a unique region, and we didn't quite know how this, based on the structure alone, we didn't quite know how this region would be contributing to hyperactivity. So we wanted to confirm that this was a bona fide hyperactivating mutation by introducing it into our in vitro enzymatic assay. This is a Michaelis-Menten curve analysis of wild type and G267S calpane 5. And this may bring back some nightmares from your undergrad biochemistry course, but we'll work through it together. You can see here that this is substrate concentration and the reaction velocity in nanomolar product per second. And you can see that our G267S calpane 5 is hyperactive compared to the wild type. we see an increase in this K-cat value, which indicates that there is increased substrate turnover compared to wild type, as well as this K-cat over Km, which represents catalytic efficiency. So then, now that we confirmed that this was a hyperactivating mutation in vitro using this substrate and a few others, we wanted to know whether or not we could take this PC2L2 loop and swap it onto a distant calpain member and see if we could see the same effect. So we took the rat calpain 1 and our PC2L2 loop from calpain 5 and swapped it into rat calpain 1 to generate this sort of Frankenstein protein. It's a calpain 1,5 hybrid molecule that's synthetic. It's not found in nature, but we can use it to test this hypothesis. So we introduced the loop into rat calpain 1 with and without the disease mutations and saw that this synthetic construct was catalytically active in vitro. And when we introduced the G267S mutation into this hybrid molecule, it's similarly led to an increase in activity. So this told us that this PC2L loop was involved in some way in the unique activation mechanism of calpain 5. And although it's poorly understood at this point, this did confirm a few things. One, that the PC2L2 loop should be studied in the context of the mutant structure, and that that the G267S mutation is a bona fide hyperactivating mutation. Now that we've identified several unique features on the capin-5 molecule, our medicinal chemistry postdoc Dylan has been utilizing these features in the design of novel inhibitors to target capin-5's function, and we should be hopefully being able to test these new inhibitors in our in vitro system soon. So we were able to learn a lot about the capin-5 structure and its function in vitro, but in order to understand the origins of adenoid disease and how a hyperactivating calpain 5 mutation would contribute to inflammation in the retina, we next needed to understand its substrates in vivo. So to give a little bit of an analogy here that I borrowed from Yasuko Ono at Kyoto University in Japan, calpains are not digestive proteases like lysozyme or trypsin. They're actually regulatory proteases that cut subdomains off their target proteins in order to activate or inactivate their function. So if we imagine protein substrates as links on a sausage, if we were to process this sausage through the lysosome system, it would grind it up into individual amino acids. If we were to process it through the ubiquitin proteasome, they would just get sliced up into oligopeptides. But if we process through the calpain system, the sausage will be cut at these individual linker domains and produce a modulated or regulated substrate with either activated or inactivated function. So in order to identify candidate retinal substrates for capin 5, we used a proteomics-based approach. We can go to the slaughterhouse and get cow eyeballs. And the reason why we use cow eyeballs is they're just large and we can get a lot of retinal tissue. We can dissect the retina from here and extract the proteins from the retina. And then we can incubate this retinal lysate with recombinant calpain 5 in the presence or absence of activating calcium, as well as a few negative controls to account for some of that background proteolysis or degradation. We can then fractionate this treated retinal lysate by SDS Page and isolate pools of proteins based on their apparent molecular weight. We can then cut out slices of this gel and isolate the proteins from these gel slices and subject them to mass spectrometry. And then once we do all this analysis, we can reconstruct this map of the gel and identify proteins that have shifted in their apparent molecular weight. And these were identified as candidate retinal substrates for calpain 5. So this is a protein-protein interaction network of 125 candidate retinal substrates for calpain 5. There were 216 identified in total. Just to orient you, each one of these circles represents an individual substrate or node, and each one of these lines or edges represents an experimentally validated protein-protein interaction between these two substrates. So 125 of these substrates were related based on known protein-protein interactions. And I went ahead and I highlighted substrates that we experimentally validated using alternative techniques. So in the interest of time, I'll only be going over one of these particular substrates, which was this one that I've highlighted here. This substrate is visual arrestin or S-arrestin. It's also referred to in the clinical literature as S antigen, and it's related to another inflammatory eye disease called autoimmune retinopathy. And patients with autoimmune retinopathy often display autoantibodies or anti-retinal antibodies against SAG and other retinal proteins. And the idea here behind this disease is that chronic inflammation, infection, cancers, or various genetic or environmental factors lead to the production of these retinal antigens that then the patient's immune response will generate autoantibodies to and start to attack these retinal proteins, which then causes inflammation leading to blindness. And it's well documented that patients with autoimmune retinopathy have anti-retinal antibodies towards SAG, as well as enolase and pyruvate kinase 2, which are all putative capin-5 substrates that we identified in our screen. So with some of the work with Kelly Schaefer in our lab, we were able to validate capin-5 cleavage of SAG in HEK cells, as well as in vitro using a series of truncation mutants. With our proteomics pipeline, we could map the cut sites of calpain-5 on SAG, and interestingly enough, two of these peptides actually correspond to known peptides that have been shown to elicit immune response in autoimmune retinopathy patients. So the idea here, or a working hypothesis, is that capin-5 that is hyperactivated in adeniv and leads to the production of retinal antigens that then causes the patient's immune response to elicit an immune response towards those retinal proteins, leading to the onset of clinical symptoms and uveitis in early adeniv. Now, I'm going to switch gears a little bit. We've been able to learn a lot about calpain-5, its structure and its function, and how it contributes to retinal disease. But at the end of the day, our team is comprised of physician scientists, and we're interested in treating these patients that are still going blind. So the question here is, how do we treat inflammatory retinal disease in adeniv patients now? And there were a few challenges that we faced in tackling this question. Again, there's no capin-5 specific inhibitor available yet, and individuals with inflammatory disease are typically treated by trial and error with various non-specific immunosuppressive medications, and adeniv patients have failed conventional immunosuppressive medications such as infliximab, which is an anti-TNF alpha antibody, as well as corticosteroids. And again, they all go blind by the time they turn 50. So our lab wanted to use a precision medicine-based strategy to treat these patients. Many of these patients undergo surgery for a variety of indications, and Dr. Mahajan and his surgical team were able to collect vitreous biopsies from these patients, and we stored them in a biobank in order to measure protein concentrations and expression in these biopsies to get some clues into the molecular causes or contributors of the disease. So we typically process our vitreous biopsies through a number of proteomics pipelines in our laboratory. I'm going to focus on this targeted proteomics pipeline that I've highlighted here. And protein concentrations are conventionally measured in these single protein ELISAs in the clinic all the time. This is routinely done, but in our proteomics test, we decided to scale this up and use an antibody-based platform that simultaneously quantified the levels of 200 cytokines. We performed this analysis on 9 adeniv patient samples, as well as 4 non-inflammatory controls. These were patients with idiopathic epiretinal membranes or macular holes. These are non-inflammatory conditions. These patients don't have adeniv, but they still had an indication for vitrectomy surgery, just because no IRB, for very good reasons, will allow us to go in and to a normal patient and collect some of their eye fluid. So we had to use these non-inflammatory controls, which were the best that we could really do in living patients. But we were able to measure differential protein expression in adeniv vitreous. Just to orient you here, this is a heat map displaying differential protein expression in adeniv vitreous compared to controls on a log scale. Orange indicates high expression, whereas green to black indicates little or no expression. And these patients are clustered according to their disease classification. So controls are clustered here, and then adeniv patients are clustered according to their increasing disease severity. And we can identify some proteins that have actual known drugs that target them. And one of the initial observations that we made that was quite striking was that there was no TNF alpha present in adenovirus vitreous whatsoever. And this explains why infliximab, which is an anti-TNF alpha antibody inhibitor, didn't affect the disease. And I want to stress that this is an important um, point to bring up when it comes to the treatment of rare diseases with precision medicine is why would you give a patient a drug if they're not expressing the drug target at all? So with this observation, we stopped giving infliximab altogether. We instead noted that patients with adenovirus had high expression of this vascular endothelial growth factor, or VEGF, in stage 3 and 4 adenovirus. This is a growth factor that promotes angiogenesis and blood vessel formation. And there's actually an off-the-shelf drug called bevacizumab, which is an anti-VEGF antibody that was originally developed for colon cancer treatment that could be repurposed for adenovirus therapy. And it's since been repurposed for age-related macular degeneration, as well as retinopathy of prematurity and diabetic retinopathy. But in intravitreal injections of this bevacizumab, we're able to clear vitreous hemorrhages in adenovirus patients without the need for surgery. So this is an example of one patient here who was only able to count fingers because of a vitreous hemorrhage blocking their vision. And following 4 weeks after an intravitreal injection of bevacizumab, they were back to their baseline vision of 20/50, which is actually quite a remarkable improvement. So we knew that steroids help to suppress the inflammation, but the use of oral corticosteroids for long-term use is not ideal because they carry a wide side effect profile that is rather undesirable. Patients get weight gain, osteoporosis, as well as other really nasty side effects. So instead, to provide high local concentrations of steroid in the eye without having a wide systemic side effect profile, surgeons are able to implant these surgical implants. One of them is called Retisert. It's this tiny implant that slowly releases steroid over the period of 2.5 years. This is a picture of my PI surgical team inserting the implant into the eye, and this is just a diagram showing where it would be inside the vitreous cavity. So the Retisert was able to suppress the inflammation and the angiogenesis, but the fibrosis in the end-stage adenovirus disease persisted. So this fibrosis was actually so severe and persistent that a fibrotic capsule would form around the Retisert implant, and then my PI and his team would have to perform this elaborate, delicate surgery to remove the fibrotic capsule and release the implant. And one of the things that we noted was after taking vitreous biopsies from patients before and after Retisert implants was that this molecule IL-6 was still elevated and was being missed by the Retisert implant. And IL-6 is a profibrotic cytokine that drives fibrosis in the setting of unresolved inflammation. So we decided to look for drugs that could target this molecule in patients that had end-stage adenovirus. So we repurposed tocilizumab, which is an anti-IL-6 antibody, for a patient that had recurrent retinal detachments. So this is an ultrasound of the eye. This is a patient that was monocular due to really aggressive adenovirus disease, and they had to have one of their eyes enucleated. So this is an ultrasound showing the detached retina in the back of the eye. This patient would undergo surgical repair for a retinal detachment and then immediately redetach following the surgery. This is an optical coherence tomography image of the back of the eye, again, showing that detached retina and some of the scar tissue that was forming in that area. So our clinical team gave monthly infusions of this tocilizumab following a retinal reattachment surgery, and the patient's retina was able to remain reattached for up to 6 months following that procedure with tocilizumab, with Retisert, with antivascular endothelial growth factor injections. So just to conclude here, personalized proteomics can guide drug repositioning efforts in the absence of a capn5 specific inhibitor. We can treat vitreous hemorrhages with anti-VEGF. We can consider IL-6 inhibition in end-stage fibrotic adenovirus. And then importantly, anti-TNF alpha therapy is no longer indicated for adenovirus patients. So just to switch gears, I think this is probably the most important part of the talk, which is the acknowledgements. So This is a picture of me, and this is the network of mentorship. So I first want to thank my PI, Dr. Mahajan. It's really been a pleasure working in the laboratory, and having the opportunity to come out to Stanford was probably one of the best decisions I've ever made. I've been able to really get creative in the lab and explore other projects that I probably wouldn't have had the opportunity to do otherwise. He's been a very supportive mentor, and has challenged me to pursue questions that are interesting, and they're sort of outside the box here. I also want to thank my thesis and comps committee, Alex Basuk, who's the chair of our committee, as well as a close collaborator of the Mahajan Lab, Ernie Fuentes, John Colgan, Steve Lenz, John Coland, who is my comps chair. I want to thank Polly Ferguson, who is a collaborator of our lab as well. I want to thank Diana Colgan, who worked in our lab for a while, and she really helped teach me how to write science very well, and helped to get me to critically think as a scientist. So I want to thank her for that as well. I owe a huge debt of gratitude to Lokesh and the protein and crystallography core at the University of Iowa. They really took me under their wing at the beginning of my PhD and taught me everything they knew about protein purification, X-ray crystallography, and structural biology. So I want to thank him, Nick Schnicker, Shankar, Paul Rogers, in the protein crystallography core, as well as the other students that helped us out, Jing Yang, Saif Khan, Hannah Koster, and Irini Petros. I want to thank Soichi Wakatsuki at the SLAC National Laboratory and Department of Structural Biology at Stanford. When we first moved to Stanford, we didn't quite have our network established for local connections and structural biology. And he took me under his wing and invited me to lab meetings and connected me with other structural biologists on campus and with resources and instrumentation that really helped us to take our structural biology research to the next level. I want to thank our postdoc, Young Ju, who's taken an active role in mentoring me in the laboratory and has been just a pleasure to work with. I want to thank my undergraduate research mentors, Raul Urrutia and Gwen Lomberg, formerly at the Mayo Clinic, but now at the Medical College of Wisconsin. I started working in their lab during my sophomore year of undergrad, and they really influenced me and motivated me to pursue the dual MD PhD training path. And their continued interest and involvement in my career development has been nothing short of appreciated, and they've been really good friends to me in the last few years. I want to thank Amy Runk at Winona State University, who is my first undergraduate research mentor, who sort of started this whole process of me getting interested in research. I want to thank the members of the Mahajan Laboratory. Here's a picture of the, I think it was a Halloween party or a Christmas party. I don't remember. But there's Young Ju. This is Jing Yang, who's our research assistant, who's helped me with a lot of protein purification and X-ray crystallography studies, as well as coding and writing different programs for bioinformatics. This is Dylan, our medicinal chemistry postdoc. This is Sarah, administrative assistant. This is Mark Terrell, who's another MSTP student in the lab, and Tasia, our clinical research coordinator, who has collaborated with us quite extensively on our proteomics and personalized medicine projects. This is Vinu's wife, Marianne. This is another picture of the lab. This is Kelly Schaefer, who is a PhD student in genetics in our laboratory, as well as Katie, who's our former postdoc. I also want to thank the Iowa MSTP, the Molecular Medicine Graduate Program, and the Alfred P. Sloan Foundation for their financial and administrative support throughout the program. I received an F30 grant through the National Eye Institute, so I want to thank them for their support as well as support from the synchrotron light sources and the Stanford University mass spec facility that helped to make a lot of this work possible. Again, I want to thank the protein and crystallography facility and all of its members and students. And I want to give a special thank you to the Stanford Ophthalmology Department, especially the chairman, Jeff Goldberg, who was very gracious and accommodating during our move to Stanford and really helped to facilitate a network for me and the other graduate students. And that support has been very appreciated in the last 3 years. Also want to thank all of my friends back in Iowa and the MSTP and all of my friends in the Bay Area that have sort of adopted me into their Bay Area family. And finally, I want to thank my brain. This is my brain. And if you look inside, you'll see that a large part of this space is devoted to music, to proteins, and to eyeballs. But most importantly, I inherited this brain from my two parents. This is Mary Bell and David Velez. I know that they're listening, and I want to thank them for their support, especially during the first few years of my graduate program, and I want to let them know that I love them very much. This is a picture of me and my family. Um, I don't know why I like this picture so much. I know we're all squinting at the sun, but this is one of my favorite pictures of me and my family. There's my mom, there's my dad, there's my brother Adrian. He looks a lot like me, as you can see. And I want to thank him in particular for, being a source of entertainment and laughter during my time in graduate school when it was needed very much. And a lot of you don't know this, but he looks so much like me that he was also able to sub for me in lab. So I was able to get away with working part-time in the laboratory while Adrian took care of the rest. So with that, I'll take any questions. If you have questions, please leave them in the Zoom chat box. If you have further questions that I didn't get to today, or if you're interested in collaborating, feel free to email me or hit me up on Twitter. And there are links to my articles here. You can scan the QR codes and it'll take you directly to those sites.